So today we will see the solved question paper of supplementary examination of 2019, April 2019 biochemistry, both 2010 and 2016 split. So first one is digestion and absorption of carbohydrate. Digestion and absorption the carbohydrate present in the diet are polysaccharide, disaccharide and monosaccharide. Polysaccharide as starch and glycogen, disaccharide as lactose, maltose and sucrose, monosaccharides as glucose, fructose and pentose. In GIT, all complex carbohydrates are converted to simpler monosaccharide from which it is absorbable form. So, digestion in the mouth. The digestion of carbohydrates starts in the mouth upon contact with saliva during the mastication. Saliva contains carbohydrate-splitting enzyme called amylase, also known as styrene. Digestion in the stomach. There is no enzyme to break the glycosidic bonds in gastric juice. However, hydrochloric acid present in the stomach causes hydrolysis of sucrose to fructose and glucose. Digestion in the duodenum. Food bolus reaches the duodenum from the stomach where it meets the pancreatic juice. Pancreatic juice contains a carbohydrate-splitting enzyme which is called as pancreatic amylase, which is similar to the salivary amylase. It requires chlorine ion for its activity. It hydrolyzes iron alpha-1,4 glycosidic linkage to get well inside the polysaccharide molecules. So, digestion in the small intestine. Main digestion takes place in the small intestine by pancreatic amylase. Digestion of completed pancreatic amylase because food stays for a longer time in the intestine. Absorption of carbohydrate has three mechanisms: by passive diffusion, precipitated diffusion, and active transport. Passive diffusion is down to concentration gradient from high to low. Precipitated diffusion down the concentration gradient from high to low, and active transport against a concentration gradient from low to high. Energy. Active transport need the energy from ATP and carrier protein required for precipitated diffusion and active transport. And speed. Passive by slowest speed and positive by fast and active by fastest mode. So dietary sources, biochemical functions and deficiency manifestations of vitamin A. Cod liver oil, liver, egg, carrot, spinach, pumpkin, butter, ghee, sweet potato, broccoli leaf, other high rich in vitamin A. The role of vitamin A in human metabolic process. Vitamin A, otherwise known as retinol, essential nutrient needed for small amounts by human for the normal functioning of the visual system, growth and development, and maintenance of epithelial cellular integrity, immune function, and reproduction. Vitamin A is essential for vision, especially for dark vision, immune response, bone growth, reproduction, maintenance of surface linings of the eye, epithelial cell growth, repair, epithelial integrity, respiratory and urinary and intestinal tract. Vitamin A also important for embryonic development and regulation of adult genes. Vitamin A and retinoic acid play a central role in development of differentiation of white blood cells, the lymphocytes, which play a critical role in immune response. The skin and mucosal cells, that is, function as a barrier and form a body's first line of defense against infection. Retinol and its metabolites are required to maintain the integrity and functioning of these cells. Keratinization of mucous membrane in vitamin A deficiency add up to the risk to the infection. Non-binding protein, a negative acute phase protein that results in decreased circulatory concentration of vitamin with a further deterioration of the immune system. Vitamin A required for general body growth, bone and brain development. Vitamin A deficiency can result from inadequate intake, fat, malabsorption or liver disorder. Deficiency impairs the immunity and hemoptysis and causes skin rashes and typical ocular effects like xerophthalmia and night blindness. Bite or spots, areas of abnormal squamous cell proliferation, keratinization of conjunctiva can seen in young children with vitamin A deficiency. Poor adaptation of darkness, which lead to night blindness, is an early symptom. Xerophthalmia results from keratinization of conjunctiva. Osteomalacia, in advanced deficiency, the cornea becomes hazy and can develop erosions, which lead to its destruction. Increased susceptibility to the infections. Keratinization of the mucous membranes of respiratory tract and urinary tract takes place. The increase in susceptibility to the infections. fatigue, anemia, diarrhea, respiratory infections, decreased growth rate and decreased bone development and infertility. Short notes. What are the isoenzymes? Give an example of isoenzyme with a clinical importance. Isoenzymes or isozymes are the multiple form of same enzyme that catalyze the same chemical reaction. Lactate dehydrogenase LD is an example of one isoenzyme. It is in a myocardial infarction, so LDH will be raised. Megaloblastic anemia, 50 times upper limit of LDH1 and LDH2. Muscular dystrophy, LDH5. And toxic hepatitis with jaundice 10 times more than LDH5. Renal disease, that is tubular necrosis or pyelonephritis, pulmonary embolism. Plasma buffers. A buffer is a solution that resists pH change upon addition of an acidic or basic component. It is able to neutralize small added acid or base, thus maintaining pH of a solution relatively stable. This is important to process and/or reactions which require specific and stable pH ranges. There are several buffer systems in the body. The most important are bicarbonate buffer, the hemoglobin buffer, phosphate buffer, proteins, and ammonium buffer. The buffer system functioning in the blood plasma include plasma proteins, phosphate, bicarbonate, and carbonic acid buffers. The kidney help control acid-base balance by excreting hydrogen ions and generating bicarbonate that helps maintain blood plasma pH within the normal range. Nearly all protein can function as buffers. Proteins are made up of amino acids which contain positively charged amino groups and negatively charged carboxyl group. The charged regions of these molecules can bind hydrogen and hydroxyl ions and thus function as a buffers. Buffering by proteins account for 2/3 of buffering power of the blood and most of the buffering within the cells. Hemoglobin is the principal protein inside the red blood cells and accounts for 1/3 of the mass of the cell. During the conversion of carbon dioxide to bicarbonate, the hydrogen ions liberated in the reaction are buffered by the hemoglobin, which is reduced by dissociation of oxygen. This buffering helps to maintain normal pH. Phosphates are found, but it is then difference between immunoglobulin G and immunoglobulin M. IgM serves as the first kind of defense, which means its first antibody to immediately develop when the foreign particle introduced, so it was temporary. This IgM found in lymph fluid, blood and produced by plasma cell. High level of IgM may give rise to parasitic infections, kidney damage, rheumatoid arthritis, hepatitis and mononucleosis. Low level IgM results in few types of leukemia and some inherited disease. IgM. Sorry, the heading has been changed. This is IgM and this is IgG. IgG is a long-term response for any disease and thus protect the body from viral and bacterial attack. Abundantly found in lymph, blood and intestine. High level of IgG in the body means long-term infections like HIV, multiple sclerosis, multiple myeloma. Low level of IgG results in kidney damage and few types of infection. Essential and non-essential amino acid. Essential amino acids are the amino acids which have to be taken to the diet and they cannot be produced by the body. There are 29 amino acids out of 20 are thought to be essential. As the definition implies, essential amino acids have to be acquired to the food such as soya, quinoa, egg, chicken, meat or vegetable protein. Serves to build and repair muscle tissue. Also, it forms precursor of molecule in the formation of neurotransmitter in the brain. High probability as the amino acids required to the food. Non-essential amino acids need not be taken to the diet as they can be produced by the body. 11 of the 20 amino acids are non-essential. They are produced within the body from other amino acids and components. Removal of toxins, intervene synthesis of RBC and WBC, promotes the brain function and immune war. Probability of deficiency is rare, but it is still occurred due to starvation or illness. Starch and glycogen. Glycogen is a polymeric carbohydrate glucose that is major component for animals and fungi. Starch is a complex sugar of glucose that is major source of carbohydrate for plants. Glycogen occurs in the form of small granules. Starch occurs in the form of grains. Function. Glycogen it serves as energy storing carbohydrate in animals, but starch serves as energy storing carbohydrate in plants. And then 2010 scheme. The first source we have already seen as a part of 2016. The first question was the same question: dietary sources, biochemical functions, deficiency manifestation of vitamin A. Second question is how heme is catabolized. The red blood cells are continuously undergoing hemolysis. process. The average lifetime of the red blood cell is 100 days. As the red blood cells disintegrate, the hemoglobin is degraded or broken into globin, the protein part, and the iron that is conserved for later use, and the heme. The heme initially breaks apart into biliverdin, a green pigment which rapidly reduces to bilirubin, an orange-yellow pigment. These processes all occur in the reticuloendothelial cells of the liver, spleen, and the bone marrow. The bilirubin is then transported to the liver, where it reacts with the solubilizing sugar called glucuronic acid. The most soluble form of bilirubin, that is conjugated, is excreted into the bile. The bile goes to the gallbladder into the intestine, where the bilirubin changes into the bile pigments. The most important one is stercobilin, which is excreted in the feces, urobilinogen, which is reabsorbed back in the blood. The blood transports urobilinogen back to the liver, where it is either re-excreted into the bile or into the blood for the transport to the kidney. Urobilinogen is finally excreted as a normal component of the urine. Urea cycle. Urea cycle, also known as ornithine cycle, is a cycle of biochemical reactions that produce urea from ammonia. This cycle occurs in the organisms. The urea cycle converts high toxic ammonia to the urea for excretion. The urea cycle takes place primarily in the liver and to a lesser extent in the kidney. Steps of urea cycle. First, carbonyl phosphatase is converted to citrulline. The catalyzed by ornithine transcarbamylase. The carbonyl phosphate group is united to ornithine and releases a phosphate group. A condensation reaction occurs between the amino group of aspartate and the carbonyl group of citrulline to form agrinosuccinate. The reaction is ATP dependent and is catalyzed by arginosuccinate synthase. Arginosuccinase undergoes cleavage by arginosuccinase to form algine and fumarate. The algine is cleaved by the alginase to form urea and ornithine. The ornithine is then transported back to mitochondria to begin the urea cycle again. Before the urea begins, ammonia is converted to carbyl phosphate. The reaction is catalyzed by carbonyl phosphatase synthase 1 and requires the use of 2 ATP molecules. The carbonyl phosphatase then enters the urea cycle. The organisms that cannot easily and safely remove nitrogen as ammonia convert it into less toxic substance urea via urea cycle, which occurs mainly in the liver. Urea produced by the liver is then released to bloodstream, where it travels to the kidney and ultimately excreted in the urine. The urea cycle is essential in these organisms because if the nitrogen and ammonia are not eliminated from the organism, it can be very detrimental or fatal. In species including birds and most insects, the ammonia is converted to uric acid or urate salt, which is excreted in a solid form. Define normal serum creatinine. The typical range of serum creatinine for adult men are 0.74 to 1.35. For adult women, it is 0.59 to 1.04. Rickets, also called osteomalacia, the softening and weakening of bones in children, usually due to inadequate vitamin D. Lack of vitamin D results in reduced absorption of calcium and phosphorus. Difficulty in maintaining proper calcium and phosphorus levels in the bone can cause rickets. Symptoms include delayed bowel aches, weakness, and pain in the spine, pelvis, and legs. Treatment include exposure to sunlight, a diet rich in vitamin D and calcium supplements, medication, or possible surgery. Two copper-containing enzymes are amine oxidase and nitrase reductase. Two cardiac enzyme markers are troponin and CKMP. IgG, immunoglobulin G is type of antibody representing approximately 75% of serum antibodies in the humans. IgG is the most common type of antibody formed in blood circulation. IgG molecules are created and released by plasma B cells. Thank you.